Okay, so am I audible to everybody? Is my voice clear? Just tell me yes or no so that I can start the class. Is my voice clear to everybody? Yes or no? Okay, fine. So I think students are yet joining the class. So we. Hmm. Okay. Hmm. So today we'll be studying about mechanisms of RNA decay and a little bit about non-coding RNA. So we have studied in our previous class in the last lecture that RNAs which are not properly formed, which are premature, which are not processed properly, in which the post transcriptional modifications are not complete and proper, those RNAs are subjected or marked for decay. Okay, so that means in order to control the transcription process, in order to control the rate of mutation so that the mutation rate is very low in our cells have devised a mechanism just like it happens in DNA replication, that when our DNA is moved from one cell cycle to another cell cycle, there are checkpoints, okay, and that checkpoints have to be passed, only then that DNA is replicated and it can be divided. So similarly here for the RNA which is not well formed, the RNA which has some problems, which is not completely post transcriptionally modified, those RNAs are subjected to degradation. And this degradation can happen and at the cytoplasm level also, or this degradation can happen in the nucleus also, in the nuclear level also. Okay, so this is just a gist of RNA decay. Now let's move to our first lecture. We have 6 lectures, so we'll try to understand the basics of each lecture very quickly because there is a lot to cover this time again. Okay, so the first lecture is mechanisms of RNA decay and non-coding RNA, that various decay pathways. So just like I told you right now that RNA decay can happen in by various ways. Okay, the first way is deadenylation dependent mRNA decay. Second is deadenylation independent mRNA decay, and the third one is endoribonuclease mediated mRNA decay. These are primarily the three main ways or mechanisms by which mRNA is decayed. So, can anybody tell me what is the meaning of deadenylation? What is the meaning of deadenylation? Anybody? What do you understand by deadenylation? Nobody knows what is deadenylation? What is adenylation? Anybody?
Removal of adenyl groups,
Yes. So
polyatate.
removal of polyatals correctly. So Kavita has very correctly told that deadenylation means the removal of the polyatal. That means the adenosine which is getting attached at the 3' end, that is getting removed. That is deadenylation. So deadenylation dependent means that it is dependent, this particular pathway is dependent on the removal of the polyatals. Independent means which is the majority of the used pathway, it does not depend on the removal of the polyatal. And the third one is the endonuclease. Now let's see what is deadenylation dependent mRNA decay. So in this what happens, there is shortening of the polyatal by deadenylation enzyme. So first there is, if this is your mRNA and this is your poly A tail. So first this poly A tail is shortened, it gets shortened, and then either there is removal of the 5' cap, either your 5' cap is removed So there is 5 prime to 3 prime decay, or because the poly A tail is removed, so 3 prime to 5 prime decay. So either of it can happen. Just a minute, someone has joined. So you need to understand that the basic of mRNA decay, I'll just explain you in this slide. This is your mRNA. Okay, this is 5 prime cap and 5 primes. There is 7 methyl guanosine cap is there, and there it is 3 prime where you have poly A tail. So as we have already understood and we have already studied in our previous classes, that if both 5 prime cap and 3 prime poly A tail is present in an mRNA, then this mRNA is very stable. That means this RNA is very difficult to degrade. So in order to degrade the RNA from either the 5 prime end or the 3 prime end, either we have to remove the 5 prime cap or we have to remove the poly A tail. Okay, suppose my objective is RNA degradation, that means RNA decay. I want to degrade this RNA. Now I have to degrade means I have to cut it from somewhere. Either I can cut it from the 5 prime end or I can cut it from the 3 prime end. Or there is one more thing that I can do. I can either cut it from between, because there is nothing, there is no security over here. Here there is one door which is 5 prime cap, here there is another door which is 3 prime cap, here there is no door. So either I can cut it from in between or I can cut it from 5 prime or 3 prime end. Just a minute, someone wants to join it seems. Yes. Okay, now if I have to cut it from the 5 prime end, I have to first remove the 5 prime cap. Okay, if I want to cut it from the 3 prime end, again I have to first remove the 3 prime cap. Only then your RNA can be marked for decay or RNA can be marked for degradation. So the first is deadenylation dependent pathway. So in this what happens, your poly A tail is shortened. Poly A tail is deadenylated. Okay, that is what is written over here, shortening of the poly A tail. And because this poly A tail is shortened, as a result of this, some other enzymes coming to picture and your 5 prime cap is removed. Your 5 prime cap is removed. Then your RNA is degraded either from 5 prime to 3 prime end if your 5 prime cap is removed, or 3 prime to 5 prime end from this side because the poly A tail has been removed. This is what is for your deadenylation dependent mRNA decay. Okay. So you can see over here, I'll just zoom it so that you can see deadenylation dependent. See, this is your this is your 5 prime UTR. Here there is 5 prime cap is there. Here poly A tail is there. Now deadenylation happens. So deadenylation means shortening of the poly A tail. Now this shortening of the poly A tail occurs with the help of enzyme which is known as CCR4 not or PARN. This is the enzyme which is responsible for the shortening of the poly A tail. Here it is written. Okay. So now what happens? The poly A tail is shortened. Now because this poly A tail is shortened, now this mRNA can be degraded by two ways. This shortening of the poly A tail has marked this RNA for degradation. Okay, and now this RNA can be degraded by two ways. First is 5 prime to 3 prime decay, and the second one is 3 prime to 5 prime decay. Now for 5 prime to 3 prime decay, what happens, that as what happens, see, for 5 prime to 3 prime decay also this poly A tail should be removed. Now because the poly A tail has been removed or shortened, now what happens, that in the place of poly A tail this LSM 1 to 7 complex comes and binds. Okay, now when this LSM 1 to 7 complex binds at the 3 prime site, it recruits another enzyme which is known as DCP1 and DCP2. It is a dimer. Two enzymes are there, DCP1 and DCP2. This dimer comes and it removes the 5 prime cap. And as the 5 prime cap is removed, another enzyme which is known as XRN1, it comes and it cuts and it starts degrading the RNA. Okay, so you can ask that if ma'am, if the poly A tail is shortened, then how the shortening of the poly A tail is related to the removal of the 5 prime cap. Even if this poly A tail is not shortened, the 5 prime cap can be removed, but it cannot happen. Why? Because for the removal of the 5 prime cap, this LSM has to bind at the 3 prime end, and for this LSM to bind at the 3 prime end, first the poly A tail has to be removed. That is why then only your LSM1 can bind to it. Okay. So did you understand? First mechanism of uh, deadenylation dependent. First, what happens? Deadenylation occurs with the help of this enzyme, and then LSM comes and binds at the 3 prime end. The binding of the LSM recruits another enzyme complex, DCP1 and DCP2, which removes the 5 prime cap. And when the 5 prime cap is removed, XRN1 comes and it degrades the 5 prime end. So this is your 5 prime to 3 prime decay. Next come your 3 prime to 5 prime decay. What happens that now the poly A tail has been removed. Now exosomes will come and it will degrade it. Okay. And exosomes will cut and chew and it will degrade the RNA from the 3 prime end. And there is another enzyme which is known as DCPS, scavenger decapping enzyme, which is again responsible for the removal of the this thing 5 prime cap. So this is your deadenylation dependent mRNA decay. So always remember that deadenylation dependent, it can occur either in two ways, 5 prime to 3 prime or 3 prime to 5 prime. In both the ways, either it is happening from 5 prime to 3 prime or 3 prime to 5 prime. Important is that this poly A site is removed. Only then LSM can bind, only then exosomes can bind. Is it clear to everybody? Deadenylation dependent mRNA decay. Is it clear? Just tell me yes or no. Is it clear or not? Because this is the basic. Okay, fine. Now, so one method of decay is deadenylation dependent. Another is deadenylation independent. Now, what happens in deadenylation independent? It means that, see, you have to understand one thing which is very important in this whole lecture, in this whole RNA decay. Some enzymes are repetitive and they will come again and again. So you always remember that XRN1, it degrades the RNA from the 5 prime end. Always remember this. That XRN1, it will degrade the mRNA from 5 prime. Decapped end means the cap is removed. And for decapping, DCP1 and DCP2 complex dimer. This dimer. Okay. So this XRN1, and this is important. It will get repeated time and again. Okay. Exosomes will are cleaving from the 3 prime end. Okay. Remember it. Exosomes from 3 prime end degradation. Okay, now you see what happens, that deadenylation independent, it is not dependent on the removal of the poly A tail. So what happens, it requires direct removal of the 7 methyl guanosine cap from the mRNA. So what happens, that this are some protein which is known as RPase 28B. So this RPS28B comes and it binds here near the poly A tail. Now, when this RPS28B comes, another protein which is known as EDC3, enhancer of decapping, it comes and it binds over here near RPS. Now, the binding of these two proteins, it it causes the decapping to take place. That means when ED3 comes and sits with RPS28B, it recruits the decapping enzyme, that is DCP1 and DCP2, decapping, DCP means decapping. It comes, it decaps it, and then again XRN1 comes and it removes the 5' end side mRNA. Is it clear? So again, same thing happening. You only have to remember that here for in deadenylation dependent, LSM 1 to 7 was coming at LSM 1 to 7, it was coming in binding through the 3' end, and that is why it needed the activity of CCR4 so that this poly A tail is removed and there is a place for LSM 1 to come and bind. But here what is binding? ED3 and RPS28B. So it can bind, and then when it binds, it recruits the decapping enzyme, 5' end is removed, and then XRN1 comes and it decaps, it degrades the RNA. This is the second one, and deadenylation independent. Then third one is endoribonuclease mediated mRNA decay. So I told you, just like here I told you, that if this is your RNA and it has to undergo degradation, so either it can undergo degradation from the 5' end or it can undergo degradation from the 3' end, but the prerequisite for degradation from either 5' end or 3' end is that this 5' cap has to be removed or this poly A tail has to be removed. This is like a bulletproof jacket. Okay, so if you wear if someone wears a bulletproof jacket, then that person is prevented from getting shot. Okay, so this is like a bulletproof jacket, so this has to be removed in order for the enzymes to attack the mRNA. But there is third way also. The third way is, what if the enzyme comes and directly targets the middle region, because there is no protection here. So that is what the third way of degradation of RNA, that is endoribonuclease mediated. So what happens? You can see many different kinds of endonuclease are there, like IRE1, PMR1, RNase, MRP. They come and they cut the mRNA from the middle. They cut in the middle. Now again, it can either go from 5' to 3' or 3' to 5'. We have already discussed that if the RNA has to be degraded from the 5' end, then XRN1 will come. If it has to be degraded from the 3' end, then exosome will come. So this is similar. The concept you should always remember. So all the three types of RNA decays, the concept remains the same. What is the concept? Again, I'm telling you, this is your mRNA, this is your 5' end, this is your 3' end. Okay, here you have 5' cap and here you have poly A tail. So RNA decay can start either from this end, which is 5' to 3', or from this end, which is 3' to, I mean, when you write it like 3' to 5', or RNA decay can start from middle. XRN1, it will degrade from the 5' end. Exosomes degrade from the 3' end. These two things remains the same. DCP1 and DCP2, this complex is required for decapping. And here, endonuclease. This is the gist of whole RNA decay. Okay, is it clear to everybody, this RNA decay? Is it clear? Yes or no? If you have any confusion, you can ask me. Okay, fine. Now, let's try to solve these two easy questions. First one is the introns are degraded due to specific enzymatic sequence, unprotected ends, lariat formation, or endonuclease activity. So there can be more than one correct option. Either of them, if you choose, it is okay. But there is one which is the uh, most important. I think someone wants to join. Just a minute. Yeah. Now this is your question. I'll give you 1 minute to think and then I'll switch the screen. Try to answer this question. Okay, I will switch the screen now. Let's see if anybody has attempted anything or not. Nobody has attempted. Why? That means you haven't understood anything. Okay. Janara is saying B. What is the B option? Okay. Anybody else who wants to try? Come on, try. Just now I have, in that slide, I have, see, this is the slide in which I told you about everything. From here only you should be able to answer the question. Here they are saying introns. We talked about mRNA, but the same concept applies to introns also. Now tell me. We have two options. Just a minute. So people are still joining, huh? So for the people who have joined just now, we have completed the lecture 1, that is the different mechanisms of RNA decay. Uh, Deadenylation dependent, deadenylation independent, and endonuclease. So if you could not understand, then you can go through the recordings, which will be available to you by today or by tomorrow morning at most, and then you can go through it to understand the concept. Okay, now we'll see the answer. So the introns are degraded due to, the correct answer is B. So both Janara and Kavita has answered it correctly. Unprotected ends. So we have just now discussed that if this is your mRNA, okay, if I want to degrade this mRNA, if this mRNA has to be degraded, it can either be degraded from the 5' end, it can be degraded from the 3' end, or we can cut it from in between also. But majority it happens from either 5' or 3' end. But there is bulletproof jacket, just like I give you an example, but there is 5' capping in poly A tail which is protecting the decay, which is making it difficult for the ribonuclease to come and degrade the mRNA. So the first and the foremost thing for mRNA decay is that this 5' cap has to be removed either, or this poly A tail has to be removed. That means mRNA or introns, they are degraded when they have unprotected ends. That means they do not have the 5' capping or poly A tailing. Okay. Now next question is, which of the following is not a step to degrade mRNA? Not. Okay. Deadenylation, 5' to 3' exonuclease activity, 3' to 5' exonuclease activity, or D, that is site specific degradation. Which is not a step to degrade mRNA? 1 minute to think and then I'll switch the screen. Try to answer, try to think. Think. Okay, I will switch the screen now. Let's see. I think people have answered. Okay, yes. So Janara is asking, ma'am, this lecture is available in YouTube or not? So yes, it is available on YouTube. This lecture would be uploaded after the completion of the lecture, and I think I have, if you can see here, if my screen is visible, so I have shared one this thing link also, and if my screen is visible, you can see. Uh, so this is the name of the YouTube channel, Purva Soni NPTEL PMRFTA. Okay, and you can see here there are lot of lectures. So there are some lectures of my previous NPTEL class also. What I'll do is that I will make a playlist. I have already made a playlist. I will update the playlist. But anyways, you can see that I have written week 5 RNA biology. So you can see from here, and you can, this is the, I think this is the week 6 class. 6 days ago. Yes, this is the week 6 class. So all the lectures, all the classes are available. Okay. And this is my YouTube channel, and I have already shared the link of one of the class. You can go and you can see. Okay. So now we'll see. I have pinned the message also. Now, so Renuka, Satyashri, Jahanara, Kavita, everybody has answered correctly. So the correct answer is D, that is site specific degradation. Okay. So we have all studied just now that deadenylation happens, and after deadenylation, either it can go for 5 prime to 3 prime cleavage or 3 prime to 5 prime cleavage. 5 prime to 3 prime cleavage means decapping is occurring. So when decapping occurs, which enzyme cleaves the RNA? At the 5 prime end, tell me, which enzyme cleaves the RNA at the 5 prime end? We have just discussed. Can anybody tell me? Okay, I'll open this slide. Now anybody can tell me? 5 prime end, which enzyme?
XRA1.
X. Yes.
XRA1.
Yes, XRN1. Sorry for my bad handwriting, but this is given in your, I mean, it is in the notes also. This is XRN1. At the 3 prime end, what is cleaving at the 3 prime end?
Exosome.
Exosomes. And which enzymes complex are responsible for the decapping of the 5 prime cap? DCP1 and DCP2. From the name itself, you can see decapping, DCP. Okay. Now we'll move to our next class, or the next lecture, that is the exosomes. So now up till here we talked about that exosomes are basically responsible for sorry, this is exosomes, so this is P bodies. Okay. Both of them are there. So, exosomes are responsible for the degradation of the RNA from the 3 prime end. Now, we'll study more in depth about exosomes. Yes, we'll study. But first, there is a small experiment that has been shown to you. Let us try to learn it. So, this week's lecture is very interesting, because nothing is getting repeated. Okay, all the slides have different concepts, just like in some of your previous slides. Some of the concept was highly repetitive, especially for your post translation modification. Things were repeated many a time, so you might get little disturbed and annoyed at what actually I have to read. But here, every slide has a different and unique information, which is actually very interesting to see. So now we'll see if you have heard about P bodies. So P bodies are what? This is extra information that I have given for you just to build your concept. So P bodies are nothing but processing bodies, and these P bodies are composed of translationally repressed mRNAs and proteins which are related to mRNA decay. That means these P bodies actually are granules. Okay, ribonucleoprotein granules. That means it has RNAs and proteins. These P bodies are granules which has RNAs and proteins. But there is a twist. What is the twist? it has only those RNAs which are translationally repressed. That means it has those RNAs which are prone for degradation, or which has to be degraded. It has only degraded RNAs. Good RNAs it does not carry the good RNAs, it carry the bad RNAs which are degraded or which are repressed, and proteins which are responsible for the degradation of RNAs. So those RNAs which are marked for degradation and the proteins which degrade those RNAs combine together to form granules which are known as P bodies. Is it clear to everybody what are P bodies? So RNAs
Yes, ma'am.
which all those RNAs that has to be degraded, plus the proteins that will degrade those RNAs. So all together they will form granules in the cytoplasm, and those granules are known as P bodies. Now, up till now we have talked about that okay, RNA is degraded, RNA is decayed. But why you think RNA will degrade at the first place? Can anybody tell me any condition when RNA is marked for degradation? Why it is such an energetically expensive process to generate an RNA through transcription, and then I am saying that RNA has to be degraded. Then why it is the need of making so much of effort of making an RNA? Why do you think that RNAs are degraded at the first place? Can anybody tell me why RNAs are degraded and what RNAs are degraded? Are all RNAs degraded? Yes or no?
No.
Okay, so then which RNAs are degraded then? If not all RNAs? Which RNAs are degraded? If not all RNAs, which RNAs are degraded?
Premature stop codon.
Okay, okay, that is also one of the
The
reasons. One
mRNA
of
has the premature.
Yes, you are right. So, one of the categories when the stop codon is not present at the correct position. So, in general, if you have to understand, so suppose in a factory, okay, so in a factory, suppose if you are making some, let's say, mobile phones, okay, suppose you have a mobile phone factory. So there you are making there is a vast production of mobile phones occurring. Okay, I'm just giving you an example for you to understand the concept. Now, after the production of mobile phones, it will undergo quality testing. Only those mobiles which have the correct quality, the optimal quality, as in all the softwares are working properly, the hardware is fine, the software is fine, everything is fine, only those mobile phones will be out for shops, for whatever, like for the people to buy. Other mobile phones or other commodities which does not qualify the quality testing, they either would be tried to repair it, or they will be discarded. Okay, it applies to any industry. So every product that is manufactured, first it undergoes a quality control. If it pass the quality control test, then only it goes further for the in the market. Similarly, if RNA is produced, it undergoes post transcriptional modifications. Then it undergoes quality testing. If the RNAs are found that they are not the optimal quality RNA, here in case how the quality is determined, either they have premature stop codons, they have they do not have the correct signals, they do not have the correct structure. So whatever the reason may be, then those RNAs are marked for degradation. So under stress conditions, what happens in normal condition, suppose if a cell is working normally, and stress condition, suppose cell is having some kind of mutation or something like this, where RNAs are now mutated, RNAs are not the optimal RNAs. So what happens under normal condition, you can see that LSM, so basically LSM and XRN, these both we have studied here. If you remember, I don't know if you remember or not, this is LSM. So LSM, it binds to the 3' end so that XRN can come and it can decay. Okay, so under normal condition, you can see that LSM, okay, I'll just, this green portion, you can see these are LSM, which binds to the 3' end. And the red portion, this red portion, you can see these are the XRN. They are wide apart from each other. They are not together. But under stress condition, sorry, under stress condition, you can see this green and yellow, green and red, it comes together, and you can see so many yellow dots are there. That means this LSM and XRN has come together, and that is why you can see so many yellow dots. Some yellow dots are here also because it is like a loop mechanism which undergoes, but you can see there are more yellow dots in the stress condition. So what does it signifies? That these yellow dots are nothing but the P bodies, that means processing bodies, where RNA along with its proteins which degrade the RNA, here they have taken the example of LSM and XRN1, they have come together. So these are your P bodies. That means these RNAs are going to get decayed. These are going to degrade. These are degrading RNAs. And here you can see that green dots and red dots are different. There are very less red dots. That means the formation of P bodies in normal condition is very less, whereas the formation of P bodies in stress condition is very more. Okay, now we'll come to exosomes. So exosomes, what are exosomes? Just now we have discussed that exosomes are required to cleave the RNA from 3' end or 5' end. Tell me, exosomes will cleave from 3' end or 5' end?
3 prime.
3' end. Very good. So exosomes cleaves the RNA from the 3' end. So that is what it is written, that these are RNA degradation complex which targets the RNA from the 3' end to the 5' end. Okay, and these are present both in the nucleus also, and they are present in the cytoplasm also. What are the function of exosomes? They are required for the degradation of mRNAs. So we already know this. And they can also degrade tRNAs, rRNAs and snRNAs. Okay, first function. Second function is that it is also required for the processing of some non-coding RNAs to functional non-coding RNAs. So exosomes are not only needed for degradation, exosomes are also required for the processing of non-coding RNAs. This is important. This function we have not explored up till now, but we might be exploring in our subsequent lectures. So, exosomes ka function number 1 is degradation, second function is processing. Now, if we look at the structure of exosomes, you see this exosome structure looks like little bit looks like a ribosome structure only. Like in ribosome, you have a smaller subunit and a larger subunit. Just like here, you can see that exosomes also have a somewhat similar structure like that of ribosomes. You can see that eukaryotic exosomes have 9 different proteins. So exosomes have structures which consist of proteins such as RRP41, and all these proteins are there. These are nothing just a reading material. I have included this slide so that you can just read and nothing much to explain. Just you have to remember the name of these proteins, because these proteins name will come further also. Wherever it is mentioned RRP some number, that means it is exosome. Okay, always remember. If anywhere in further slides you will find RRP41 any number, RRP, that means it is a protein of exosome. Now you see how exosomes regulate. So just now we saw that exosomes are responsible in both. It is responsible in processing of mRNA, processing of non-coding RNAs and degradation. And it functions both in nucleus and cytoplasm of the eukaryotic cells. Always remember two things. So whenever we are talking about exosomes here, it functions both in nucleus and cytoplasm. One thing. Second thing is It is responsible for degradation also, and processing also. Okay, these two are important. So they have such a diverse function, exosomes have such diverse function, there are a number of different proteins and protein complexes. The two most studied example are tramp and SKI. So always remember, as now just we discussed, that exosome can perform its function in nucleus also and in cytoplasm also. So in nucleus and cytoplasm, the environment is different. How the environment is different? Can anybody tell me? What is the difference in the environment of nucleus and the cytoplasm? Anybody? Anybody knows how the environment of a nucleus is different?
Nucleus is basic.
Okay, yes. Yes, okay, so this is also correct. And one very important point which applies, which is the basis of a lot of concepts that we study in molecular biology, is that cytoplasm has a reducing environment. Okay, and because cytoplasm has a reducing environment, that is why when your mRNA it comes out of the nucleus, and because it has a 5 prime cap, it has a 3 prime tail, so the mRNA is protected. And in the case of prokaryotes, because there is no nucleus and cytoplasm compartmentalization, that is the reason why transcription and translation occur simultaneously. Because when translation is also occurring and transcription is also happening to the growing mRNA chain, the ribosome gets bind because for doing translation, and the binding of the ribosome not only is responsible for translation. Remember, the binding of ribosomes also protects the mRNA, because mRNA is present in the reducing environment only. In the case of eukaryotes, your mRNA is protected up till it is modified because it is inside the nucleus. Once it comes out of the nucleus, it has all the protections in the form of 5 prime capping and tailing. But in the case of prokaryotes, there is no such protection because there is no nucleus. That is also a reason that our body has devised a mechanism that we will do translation and transcription simultaneously. So translation will also occur and ribosomes will in a way protect the mRNA also. So what I'm trying to say, that because the environment of nucleus and cytoplasm is different, therefore the exosome complexes that are required in nucleus and cytoplasm, they will also be different. So in nucleus we require tramp. Okay, nucleus there is tramp complex, and in the cytoplasm it is sky. So you can see here, tramp complex, it consists of air 1p, air 2p, and all these things. And sky complex consists of these three things. So what these are just the the proteins which are there in the two complexes. Important thing to understand is that tramp complex is responsible in the nucleus and sky complex is responsible in the cytoplasm. Okay. Now we'll come to lecture number 3, which is actually the continuation of what we are studying just now. So now we'll see how the tramp complex does its work. Okay. See, you may think that, oh God, there are so many mechanisms, so many protein names that I have to learn in this particular, it's getting a lot. But see, it's very interesting if you try to understand the logic and concept behind this. Okay, you might have to remember some of the protein names here and there, but if you know the concept, you can easily identify the name. Especially in MCQ, it is easy because the answer is there in front of you. Even if you remember just a little bit about the name, you can easily identify it. Okay, so it's easier in MCQ to pick the option, in my way. And the concept, the logic remains the same. The logic is same, you just have, there are just some modifications that are happening. Here and there, some few modifications are happening. So, anyways, so now we were talking about that exosomes have many different complexes. One such complex is the TRAMP complex, and this TRAMP complex works in the nucleus. Now we will see how this TRAMP complex works in the nucleus. So why this TRAMP complex works? For the degradation of aberrant RNAs in the nucleus. Aberrant means improperly processed. Those RNAs which are not properly made. If these RNAs are improperly made, so what do you think that improperly processed RNAs will lead to if they are not degraded? Suppose if there is an RNA, it is not made properly, it is not processed properly, and it is not degraded. Then what will happen according to you? Can anybody tell me? What will happen if RNA is not processed properly and it is not degraded? Somehow it skips the degradation process. Try to answer. Nobody? Hmm? See, two things can happen. First, because it is improperly processed, so it will not make its respective protein, or it will make a protein which is not functional, or it will make a protein which is functional but it will code for some different function, and that different function can be very detrimental for the cell, for the body. Okay, so that is why in either of the case it is not a good condition. It might lead to some kinds of diseases, it might lead to some kinds of pathological conditions. Therefore, those RNAs that are improperly processed, that means aberrant RNA, have to be removed from the nucleus. And the removal of those aberrant RNAs from the nucleus happens with the help of exosome, in particularly the TRAMP complex. Okay, now, how to identify an RNA which is improperly processed, which is an aberrant? So, an aberrant RNA which is improperly processed will have a very slow 5' capping and a very slow poly A tailing also. Okay. So, always remember, a RNA which is malformed or RNA which is not proper, it will have a very slow polyadenylation, very slow capping will take place. So, you can see here, this is your TRAMP complex. The TRAMP complex comprises of these three proteins. So the TRAMP complex, it trims the poly A tails of RNAs which are destined for RRP6, means which are destined for exosomes and the core exosomes. Okay. So basically what happens, that this TRAMP complex will cleave the poly A tail up till 4 to 5 adenosines. After that, whether cleavage will occur or not, it depends upon whether the RNA is aberrant or not. If the RNA is not aberrant, if the RNA is not bad, then the polyadenylation would be very fast. If the RNA is improperly made, if the RNA is improperly processed, then polyadenylation would be very slow. Okay. Now what will happen? The RNA in which polyadenylation is very slow, that is not a good RNA, that is a bad RNA. And that RNA would be degraded. And the RNA in which the polyadenylation is fast, that means it is properly made, it is properly processed. So there, what will happen? That RNA would not be degraded. So whether the RNA would be degraded or not, it will depend whether polyadenylation is happening fast or not. Is it clear to everybody? If polyadenylation is happening fast, that means it is a good RNA, no need of degradation. If polyadenylation is happening slow, that means a bad RNA, it has to be degraded. Now, so this we have studied uh, in the nucleus. Now in the cytosol, now comes different. See, it might look a little difficult to you, but if you are giving your 100% attention, then it is easier for you to understand, and when you will get back to the recorded lectures, things will become much more easier. For those who have already been through the lectures, already saw the lectures, and if you're coming here, that means you are reading the chapter for the second time, so things would be clearer to you. For those who are reading all these things for the first time, I request to again go back to the recorded lectures, and when after listening here, again when you will listen there the recorded lectures, things will become much more clear to you. Okay, so up till now we have studied that in the in the nucleus, this tramp complex helps in the degradation. Now in the cytosol, this protein is responsible, sky protein is responsible, that we have learned. But there are different mechanisms altogether in cytosol that happens. What are the three mRNA decay mechanisms that happens in the cytosol? Now remember, everybody, please pay attention. This is important. Okay, this these three mechanisms: non-stop mediated mRNA decay, non-stop mRNA degradation, and nonsense mediated mRNA decay, non-stop mRNA degradation, and no-go mRNA decay. These three mechanisms are important for an exam point of view, and in general, even for competition per se, these topics are, I mean, these three mechanisms are important. Questions might come from this part. So I have already told, I think in some of the class, I don't remember the exact class, but I've told about nonsense mutation. So can anybody tell me what is a nonsense mutation? What do you understand by a nonsense mutation? Okay, so I'm going to pick myself. So Kusuma, can you tell me what is a nonsense mutation, please? Any idea? We have already discussed this thing in the class. Nonsense mutation? Anything that you know, anything. It's fine. We are just discussing. We all are learners. We all are learning. We are just sharing our knowledge with each other.
Mutation occur in nonsense codon, madam.
And what are nonsense codons?
Stop.
Stop codons. Yes. So you're correct. So basically, when mutation occurs in the stop codons, then the protein which is formed is a truncated protein. So that means if the protein has, let's say, 600, if the length of the protein is 600, because of the nonsense mutation, it might come down to, let's say, 250. So this is a nonsense mutation where mutation occurs in the stop codon. The mutation occurs which leads to the formation of a stop codon. That is, if suppose you have, what are the stop codons? UAA, UAG, and UGA. Suppose there is a codon which is GGA, and there is a mutation in G, and now it is converted into U. So now you have a UGA. So earlier it was GGA, but because of this mutation, G is converted into U, and now you have UGA. So now when this UGA will be encountered, your protein production would stop. So you will have a protein which is truncated. That is your nonsense mutation. So if this is your nonsense mutation, then nonsense decay means that target mRNAs that contain a premature termination codon. So if your mRNA is formed in such a way that it has a premature termination codon, then that is your nonsense, then that mRNA is marked for degradation, and that kind of degradation is known as nonsense mediated mRNA decay. Then non-stop mRNA degradation means non-stop. target mRNAs that do not contain a stop codon. Non-stop, it is going to translate only. That is also wrong. So premature termination of translation is also wrong, is also not required, and continuous translation is also not required. That means protein or amino mRNA should be translated up to the optimal length, not less than that, not more than that. Either of the case is detrimental for the cells. So, if the target mRNA has a premature termination codon, that means that RNA would be of a lesser size. That is also not a good condition. If your target mRNA does not have a stop codon, it will go more than its desired size. That is also not a good condition. Then no go mRNA decay. No go means that your ribosome is there, but it is not able to go. It is not able to move. Ribosome is stalled because there is a secondary structure. So, if this is your RNA, there is some kind of secondary structure. So, if this is your ribosome, this ribosome is not able to move forward because of the secondary structure. So it is not able to move, not able to go. This is no go. And why these all are, why they all occur in the cytosol? Can anybody tell me why all these are in the cytosol only? Why these nonsense mediated decay, non-stop and no go, these happen in the cytosol? Why can't they happen in the nucleus? These kinds of decay cannot happen in the nucleus. Why? Tell me. Why nonsense mediated decay or non-stop mRNA degradation or no-go mRNA decay, it happens only in the cytoplasm and not in the nucleus? Tell me. Think. It's very easy, yeah. It's very easy. You just have to put your mind into it. In all the three kinds of, nobody, anybody wants to try? Renuka Devi, Yuvaranjani, Kavya, Rukmini, anybody, Kanchana, Swapna, Trishala, Teja. I hope you all are here and not just have kept your lecture and went somewhere else and doing something else. I hope that this is not what you are doing. You all are listening to the lectures. Okay,
Because translation process takes place in cytoplasm.
exactly. That is what is the correct answer. See, very good. So you see all these decays, nonsense, non-stop, no-go. There, we all in these all the three types of decays, we are targeting the translation process. It is related to translation process. Premature termination codons. There is no stop codon. It is that means all the three are related to translation or ribosomes, and translation occurs in the cytoplasm. So this cannot happen in the nucleus because they all the three are somewhat related to the process of translation. They are related to ribosomes. Very good. So that is why these kinds of RNA decay, mRNA decay cannot happen in the nucleus. They will happen in the cytoplasm only. Fine. Now let's move. So now we'll see the nonsense mediated mRNA decay. This is just for your information. Say I have included this screenshot. Nothing much to explain here. You can just read through. Okay. So nonsense mediated decay. So now this is eukaryotic release factor E. Okay, just a minute. ERF3. ERF3 is eukaryotic release factor 3. Okay, this is responsible for the release of the ribosomes after the translation is completed. If you have studied about translation in eukaryotes, you might have read about this particular factor. Now, in normal cases, because it is a release factor, it will bind to poly A binding protein. That means the translation is completed. Now, poly A binding protein has to come and poly A tail has to be added. But what happens in certain cases, it does not, poly A binding protein is not there, or there is UPF1 is there. And when UPF1 is there, then it will bind to UPF1, and then what happens? It undergoes nonsense mediated mRNA decay. Okay, so ERF is looking for poly A binding protein, but if it finds UPF1, then that is a sign that it will go for nonsense mediated RNA decay. That means before the binding of poly A binding protein only, UPF1 has come. Now this UPF1 has come, so now there is a competition whether PABP will bind or UPF1 will bind. And UPF1 wins and your ERF binds with UPF3 and the mRNA is degraded. So there is no binding of poly A binding protein, nothing happens, mRNA gets shorted. Very easy. ERF should bind to PABP, but if it encounters UPF1, there is a competition between the two. UPF1 wins and ERF binds to UPF1. This is your nonsense mediated mRNA decay. Now, very simple question we have studying about all this all the way long. TRAMP complex regulates exosomes in the cytoplasm. True or false? 1 minute to think and then I'll switch the screen. Although it should take hardly 30 seconds. Think. Okay, now I'll see what students have answered. Okay. So Jahanara has said true, Kavita has said false, Renuka has said false. Okay. Okay. So the correct answer is false. Why? Because TRAMP complex operates in the nucleus. We have just studied here. See, in the nucleus, TRAMP complex come. TRAMP in the nucleus, sky, see, TRAMP is in the nucleus, SKIY, sky in the cytoplasm. Always remember. So there are many different complexes of exosomes. So if I make it like this for you, for your basic understanding, okay, see, exosomes, it can operate in the nucleus or in the cytoplasm. In the nucleus with the help of TRAMP complex, in the cytoplasm in the help of ski complex. Again, in the cytoplasm, I'm sorry, but my handwriting is not good, especially in, you have nonsense mediated decay, non-stop mediated decay, and no-go mediated decay, in which we have studied about TRAMP and we have studied about nonsense mediated decay. Okay, is it clear to everybody? Yes or no? Tell me. If there is any problem, tell me. Is it clear? Okay, fine. So now we'll come to, so this is just another, you can again see nonsense mediated decay, that there is a stop codon here. The stop codon is present premature. I mean, it should not be present here, it should be present at the end, but there is a premature stop codon over here. Now, whether it will uh, be marked for degradation or not, it depends on the distance. If the distance of the stop codon with the exon junction is more, then that means that some editing has occurred post transcriptionally, and that is a potential nonsense mediated target. What does it mean? That in an exon, the stop codon should be present at the end. Okay, but suppose what happens is, we have studied about differential splicing, differential expression of genes. So sometimes, if you, I don't remember, I don't know if you remember or not, we have studied an example where a same protein, it is of different length in one organ and a little longer in another organ. So that means one protein, if suppose the maximum length of one protein is 600, it can be present as 250 in length in lungs and 600, let's say, in eyes. That means all the proteins are expressed in the eyes, but not all the proteins are expressed in lungs. In lungs only 250 proteins are expressed, I mean amino acids are there, and in eyes only 600 amino acids are there. So I mean all 600 amino acids are there. So this means that the differential expression of genes can also regulate the length of proteins. Now, how can you differentiate whether the stop codon that has been incorporated, it is because of the differential expression, or it is something wrong has happened, that is why this stop codon has incorporated? Do you understand my point? Because it can be the case that stop codon has incorporated so that the differential expression of gene can take place. So how can you differentiate between the incorporation of these two kinds of stop codons? So you can differentiate by the distance. That means if the distance of the stop codon from the exon junction is more than 55 nucleotides, that means this stop codon has is not a default stop codon. This stop codon has occurred post transcriptionally, and therefore it has to be decayed. Whereas if the distance of the stop codon from the exon junction is less than 55 nucleotides, this means that this stop codon was present before transcription. That means it is regulating some differential gene expression, therefore it is not mediated for a nonsense mediated decay, not targeted or not marked for nonsense mediated decay. Okay, is it clear? Is it clear or not? What I'm trying to tell is clear or not? That stop codons can be incorporated during transcription so that same mRNA can code for different proteins. We have studied about differential expression of genes. But to differentiate such stop codons from the malfunction stop codons, the distance has to be considered. If the distance of the stop codon from the exon junction is more than 55 nucleotides, it is a nonsense mediated decay target. If the distance is less than 55 nucleotides, it is not a target. It is correct. So this is just another image for you to see. I've told you that endonuclease will come, and then at the 3' end exosome will come, at the 5' end XRN will come. This is what we have already studied. Now we come to non-stop decay. So non-stop means non-stop the translation is going on, going on, going on. Why the translation is going on? Because either stop codon is not present or ribosome is not getting stopped. Both the reasons can be there. Stop codons is not there, or ribosome is not there, or ribosome is not getting stopped. Ribosome is continuously moving and it is continuously synthesizing. Okay, so this is known as non-stop decay. So that means those mRNA which are translating non-stop, how they are decayed? So here comes your ski proteins. Ski proteins that we have talked about now. Earlier we talked about these ski proteins. So here comes your ski proteins. So what they do is that ski protein comes and they binds. Okay. And what happens? These ski proteins, they recruit the exosomes, and exosome causes the 3' to 5' decay. Or if there are no ski protein, then what happens? Decapping occurs, and then 5' to 3' decay occurs. So in non-stop decay can happen either in 3' to 5' manner or 5' to 3' manner. This we have studying repetitively so many times. So for 3' to 5' decay, what happens? Exosomes comes. We all have studied that if the RNA has to be degraded from the 3' end, exosomes has to come. So these ski proteins, they bind to the 3' end, and the binding recruits the exosomes, and exosomes cleaves. Or decapping occurs and XRN1 comes and it cleaves the 5' end. So this is your non-stop decay. Next is no-go decay. So no-go means ribosome is stalled due to RNA secondary structures. So I have included these small lines so that when you go through this thing slides, at least you have a little bit of context of what is happening in the picture. Otherwise, because there are so many mechanisms, so many pictures are there, that you might get a little confused. You might understand right now, you might feel that you have understood everything, but when you will again go and when you will try to recollect, then you might get confused because there are so many mechanisms. That is why I've just included one line over here so that you can go and just read it. Okay, so here what happens ki ribosome is stalled. So when ribosome is stalled, see, yahan pe you can see there is a strong RNA secondary structure is there. And because there is a strong RNA secondary structure here, the ribosome is unable to move. And because the ribosome is unable to move, then some protein which is known as DOM34 HBS1, it comes. This signals that endonucleatic cleavage has to be there. So endonucleatic cleavage occurs, and then 3' and exosome comes, 5' and XRN1 comes. These two are important. That's why at the beginning only I told you that remember that exosomes will attack the 3' end and XRN will attack the 5' end. This you have to remember. Okay. Next. Now we'll try to solve some questions. Just a minute. And then we'll move to lecture 5. So up till here, is it clear? Is there any confusion? You can ask me. If there's any confusion, just ask me. Is it clear? Yes or no? Is it clear? No one is answering anything. Okay, I assume that it is clear if nobody is answering. Okay, no go decay once. Okay, fine. So, see, this is your mRNA, single stranded mRNA. So, whenever your translation happens, your mRNA is single stranded. Only then your ribosome is able to move over the mRNA. This is your ribosome. It is moving. Very happily, it is walking over the mRNA. So, all of a sudden, there is a secondary structure. For example, you are walking on a road. Happily, you are walking on a road. What happens? Suddenly, you see that the road is broken. There is a bridge, and the bridge is broken. The road is broken, and you cannot move further. So, you are stalled at this particular place. You cannot move further because the road is broken. Similarly, it happens with the ribosome. The ribosome is happily moving over the mRNA, but there is secondary structure formation. So, this is loop. This is your loop. This is a kind of secondary structure that happens in the mRNA. Now this secondary structure has come. Now this mRNA cannot move. So what will happen? Sorry, this ribosome cannot move. So now this ribosome is stalled. That means ribosome is not moving. It is just at its place. Now what happens when such situation comes? Then no go decay happens. So a kind of decay where ribosome is not able to go, where ribosome is not able to move. Then what happens in such situation? The stalling of the ribosome creates a signal that this mRNA has to be cleaved endonucleotically. So endonucleotic cleavage occurs with the help of DOM34 and HBS1. They will come and they will bind and they will cause the endonucleotic cleavage. When endonucleotic cleavage occurs, then this will become 5 prime, this will become 3 prime, this will become 5 prime, this will become 3 prime. That means now this particular this thing mRNA is broken into two parts, and each two parts will have its own 5 prime 3 prime and 5 prime 3 prime end. Then in 3 prime end exosome will come and it will degrade the mRNA. In the newly formed 5 prime end xRNA will come and it will degrade the mRNA. Is it clear or not?
Yes, ma'am.
Okay, so Singhi Reddy, Gaurishwari, is it clear to you? Okay. Okay, now we'll move further with the questions. Mm. Nonsense mediated RNA decay occurs when RNA encounter a stop codon, RNA lacks exon junction complex, RNA lacks poly A tail, or RNA has a premature stop codon. Well, 1 minute for you to think and then I'll switch the screen. Okay, let's see. Oh, sorry. Yeah. So, Janara has answered D. Anybody else who wants to try? What should be the answer? Nonsense mediated RNA decay occurs when? Okay. So Kusuma has also answered D and Satya has also answered D. Okay. So D is the correct answer. So we have already studied that stop codon has to be there. But if it is present premature, that means it is present at a position very before then it should actually be present, then your RNA would be a truncated one. So that RNA is degraded and that is your nonsense mediated RNA decay. Then non-stop decay refers to RNA degradation when protein synthesis do not stop, there is no stop codon, there is premature stop codon, intron is present. Again, very easy question. Well, okay, now I'm switching the screen. Okay, so Janera has answered B, Kavita has answered B, Satya has answered B. Let's see what is B option. There is no stop codon. Non-stop decay. Yes. Correct answer. That means now you are understanding the concepts. Good. Now, next question is no-go decay occurs because of incomplete splicing, alternative splicing, absence of 5' cap, or unique RNA secondary structure. 1 minute and then I'll switch the screen. Okay, I'll switch the screen now. Okay. So, Gaurishwari D, Renuka Devi D, Kusuma D, Jahanara D. Okay, correct. So the correct answer is D. No go means the ribosome is unable to go. Ribosome is unable to move. And why ribosome is unable to move? Because in between, ribosome has encountered some kind of secondary structures. That's why ribosome is unable to move. So no go decay. So this non-stop nonsense, it refers to the condition of the RNA. Nonsense means because there is a premature stop codon. Non-stop means there is no stop codon. No go means not able to move because there is secondary structure. Then non-stop mediated decay is observed mostly in bacteria, fungi, protozoa, animal. Okay. Now I'm switching the screen. Okay. So Satya, Janara, they have answered D. Anybody else wants to try? Fine. So the correct answer is D, animals, because whatever we are studying about eukaryotes. So it is animals. Okay. Now we'll move to lecture 5, which is auto-regulation of RNAs. So now we have studied about uh, up till now we were studying about the RNA decay and the different mechanisms of RNA decay: deadenylation dependent, deadenylation independent, endonuclease dependent. Then we studied about exosomes, how exosomes regulate the decay of RNA. We saw that exosomes can regulate the decay of RNA inside the nucleus through TRAMP complex, or inside or in the cytosol through three different pathways, that is nonsense mediated decay, non-stop mediated decay, and no-go decay. And now we are studying about the auto-regulation of RNAs. So in auto-regulation of RNAs, there are many different examples that are given. So I've tried to incorporate all the examples. Maybe one or two examples are not there. So I just will quickly we'll see how it happens. So you see here RRP6. What is RRP6? Can now tell me what is RRP6? We have 6P. I have already told you like somewhat back that you might encounter this term time and again in this lecture. So tell me what is RRP6? What is RRP6? Where we studied this RRP6 today in our class? Do you remember? Where we studied RRP6? Nobody remembers? See here, when we were studying about here, I told you now that these are the different kinds of protein structures or different kinds of complexes which are present in exosomes, you will get RRP. See, RRP, if RRP is coming, that means it is a protein of exosome. Always remember this RRP term might come again and again. Okay. So, yeah. So what is written over here? That this is a particular mRNA which has RRP6. This RRP6 is nothing but a protein of exosome. So you just consider this RRP6 as exosome. Now this RRP6, it cleaves the 3' site, 3' poly A tail, that we know. Now, this 3' poly A tail, which is cleaved by RRP6, is dependent on the concentration of a protein, which is NAB2, which encodes poly A binding protein. Just a minute. Okay, fine. So, what happens if poly A binding protein, that means if poly A tail is there or not, and what is the concentration, it depends on that. I'll explain you. So this RRP6, it will start cleaving the poly A tail. It will cleave, and it will cleave up till 26 A's are left. Okay. So what happens if this is your 3' end? You have A1, A2, A3 up till A26, and then you say you have A200, and here you have RRP6. RRP6. Now this RRP6 will start cleaving the A's. It will cleave. It will stop. That means up till 26 A's, when 26 A's are left, then RRP will stop. Now whether it has to cleave all the remaining 26 A's or not, it will depend on the concentration of NAB2 protein. If NAB2 is low, then what happens? There is no degradation. What does it mean? If poly A binding protein is already present, if poly A binding protein which will make poly A, which will make the poly A tail, it is already present in excess, that means this A can be removed, then only this NAB we can add more and more poly A tail. But if this NAB2 which will code for poly AB is very low, then if this RRP6 will cleave these 26 A's also, then nothing would be left. So that is why the trimming will depend upon the concentration of NABB2. If it is low, then it will not cleave it. If it is very high, then it will cleave it. So this is the auto regulation of NAB2. When NAB2 is high, that means NAB2 is already present a lot, then it will cleave the poly A tail. When NAB2 is very less, that means not much poly A tails can be cleaved, then it will not cleave the poly A tail. So this is the auto regulation of NAB2 which is happening. Okay. Then we come to degradation of meiotic transcripts in vegetative cells. Now, what is the meaning of this? So basically, you understand that there are two types of uh, cell divisions that occurs. First one is a mitotic division, and second one is a meiotic division. So mitotic division occurs in mitotic cells, which are also known as vegetative cells, and meiotic divisions occur in meiotic cells. Now the transcripts, just like we have studied, that there is a differential expression of genes that happens. Okay, so differential expression of genes means that genes are expressed in a cell specific manner. The genes which has to be expressed in the lungs will not be expressed in the eyes, and vice versa. That means those transcripts that has to be expressed in the meiotic cells should not be expressed in the vegetative or the mitotic cells. So how this is regulated? This is regulated with the help of this mRNA degradation. That means the meiotic transcripts, that means those transcripts that will code for meiosis, those transcripts are degraded in the mitotic cells or vegetative cells. How it happens? This is the mechanism. So how this happens? So basically DRS, what are DRS? So DRS containing RNAs are useful in maintaining the meiotic transcripts. That means in the mitotic cells, DRS containing RNA, it has to be degraded. Okay, in mitotic cells, DRS containing RNA has to be degraded. So how it is degraded? See, in vegetative cells, when DRS is bound by MMIP, this DRA is bound by MMIP, it causes exosome recruitment that eventually leads to degradation, and hence RNA is degraded. That means this MMI1P, this is responsible for DRS RNA degradation. Okay, so in mitotic cells, what happens? This MMI1P, it comes and sits on DRS and it recruits the exosomes, and exosomes cleaves the RNA. But in meiotic cells, what happens? That these MMIP1, what happens? These MMIP1s, first all the MIP1s come together, and then another protein which is known as Mi2 dots, this comes and they remove the whole cluster of MIP. So MIP is not able to bind the DRS sequence, and hence no degradation takes place. Is it clear? Is this mechanism clear to you or not? Tell me. This mechanism of degradation of meiotic transcripts in vegetative cells. Is this clear, or if there is any confusion, please ask me.
Explain once more.
Okay. See, so basically you need to understand that certain we know about differential gene expression. That means the transcripts are expressed in a cell specific manner. So those transcripts, all the genes are present in all the cells, but all the genes are not expressed in all the cells. So similar concept applies here also, that transcripts or mRNA or RNA, which is responsible for meiosis, it should not be expressed in the mitotic cells. Okay, it should only be expressed in the meiotic cells. So how this is controlled? So this is a transcript, this is a RNA transcript which ultimately helps in meiosis. This DRS containing transcript, it helps in meiosis. So this transcript should be degraded in the vegetative cells. Vegetative cells means mitotic cells. Okay, this transcript should be degraded in the vegetative cells. So how this is degraded? This is degraded because this meiosis containing transcript has one region which is known as DRS region, and in this DRS region one protein, the name of the protein is MMI1P, it comes and binds, and when this MMIP protein comes and binds to the DRS region, it recruits exosomes, and exosomes degrade this RNA. So this meiotic transcript is not expressed in your vegetative cells, but we want this meiotic transcript to express in the meiotic cells. So what happens, that all the MMIP which is present in the meiotic cells, they all first come together, all forms a cluster, and then this whole cluster is removed with the help of another protein which is known as MEI2 dots. So this dot protein removes the whole cluster before it can bind to the DRS. So before this MMIP comes and bind the DRS, this Mi2 dots, it remove this cluster. And hence this DRS is prevented from degradation. That means DRS containing RNA can be degraded when MMIP1 protein comes and it binds to DRS. If by mechanism we can prevent this MMIP to bind to DRS, then it will be activated, and that is what is happening here. This MEI2 dots, they are actually removing this MM1P from the cell so that it cannot come and bind to the DRS containing RNA. Is it clear? Is it clear to now? Is it clear now?
Yes, ma'am.
Okay, so Kavita, is it clear to you?
Yes.
Okay, fine. Now, next is again, so there are so many mechanisms. I think this is the last mechanism, no? Yeah. So again, another thing is exosome and gene expression. So now here they have talked about the comparison of young cells and aged cells. So what happens in young cells, this exosome, which is RRP6P, it is very active. So it can do an antisense approach. That means it can degrade the antisense RNA. So what happens in young cells, there is one RNA which is a 481 RNA, 484 RNA. This is 484 RNA which is present in young cells also, and this RNA is present in old cells also. But what happens, this 48 RNA has a sense strand and an antisense strand. So what happens, that RRP6, it degrades the antisense strand of this 484 RNA. Whereas in aged cells, what happens, the RRP6 activity is very low, so antisense RNA of the 484 is not deleted, and therefore the activity of this RNA is very less. And see, I'll tell you what happens, what is happening over here. See, you have young cells and you have old cells. So basically, this 484 is a particular RNA which is very much expressed in the young cells. And the expression of this RNA occurs only when the antisense part of this RNA is deleted, because RNA works only when it works best when it is single stranded. When it is double stranded, then RNA cannot work. So young and old, in young cell, this 484 is very much activated because the antisense portion of this RNA is deleted. Whereas in old cells, this 484 is not active because it has both sense plus antisense strand. Because it is in a double stranded form, so it cannot function properly. So in young cells, this 484 is very much expressed because the antisense RNA means 484 has actually a sense RNA and an antisense RNA. Both are there in 484. Okay, this antisense portion is deleted in young cells because of the activity of exosomes. And what is the name of this exosome? You can see over here, the exosome is RRP6C. So this RRP6, it deletes the antisense portion of 484, and therefore 484 is very much active in the young cells. Whereas in the case of old cells, this RRP6 activity is very low. Therefore, it cannot delete the antisense portion of the 484 RNA, and therefore 484 RNA is not active in the old cells. So this is what is there, that the expression of exosomes can control the expression of RNA also. Because RRP6, this exosome is very much expressed in the young cells, therefore it is able to degrade the antisense portion of this RNA, and therefore this RNA is expressed a lot. Whereas in old cells, in aged cells, because this exosome RRP6P is not expressed, therefore, I mean, this activity is very low. Therefore, the antisense portion is not deleted. Therefore, the expression of 484 is very less. This is how the expression of or the activity of exosomes, it regulate the expression of RNA. Is it clear? How the activity of exosomes is regulating the expression of RNA. Okay. Now, now we'll come to the last lecture. So last lecture, I think there is not much given in the last lecture, because probably we'll study about non-coding RNA in the subsequent classes. So what is a non-coding RNA? A non-coding RNA is a functional RNA that does not translate into a protein. That means those RNA that do not make a protein, but still they have some role. It is not that non-coding RNAs do not have any role at all. They have role, but they are not translated into protein. They do not code for any protein. Now there are two main class of non-coding RNA. First are housekeeping non-coding RNA, and second is regulatory non-coding RNA. And what are housekeeping non-coding RNA? Those non-coding RNA which are expressed all the time and which are responsible for normal function. Just like we have, if I don't know how many of you, if you have done some real-time PCRs or some Western blotting, there are certain housekeeping genes, like you have beta actin or GAPDH. So those housekeeping genes are the genes which are active all the time, which are constitutively expressed. Similarly, there are some housekeeping non-coding RNAs which are expressed all the time. And then there are some regulatory non-coding RNA, and regulatory non-coding RNAs are the RNAs which are expressed in certain stages or in response to certain stimuli. That means regulatory non-coding RNAs are not expressed all the time. They are expressed in certain stages. Those are regulatory non-coding RNA. Okay. Then there are certain examples of housekeeping RNA. You can see there are so many housekeeping RNAs are there. tRNA, rRNA, siRNA, snRNA, guide RNA, telomerase RNA. And then there are other also like speckles, paraspeckles, fragile bodies, genes. These are all the examples of housekeeping non-coding RNAs. Then again, this is not very important, just to show that the signal recognition particle SRP, it is also composed of two kinds of RNA, which is 4.5s RNA and 75s RNA. Just to show, this is just an example to show that non-coding RNAs, although they do not code for a protein, but they still have very important functions in our body. For example, the signal recognition particle is composed of two kinds of non-coding RNAs, 4.5s RNA and 75s RNA. Okay, now we'll solve some questions quickly. Abolishment of meiosis in yeast occur when there is no nutrition, when DRS containing regions are degraded, when MI dots are absent, when DRS containing RNA are stabilized. I'm giving you time, 1 minute to think. Tell me. So Renuka has said B. Okay. Others? Let's see if others can also give me some answer. Jarara has said B. Waiting for others to respond as well. I'm giving you 1 minute and then I will switch the screen. Okay, let's see. I'm switching the screen now. Sorry. Okay, so I have just two answers. Okay, fine. So the correct answer is B only. How? Let's see. So for meiosis, we have studied that DRS RNA is responsible for meiosis. And this RNA is degraded in mitotic cell. This is what we have studied. Now, this is responsible for meiosis. So if there is abolishment, means what is the meaning of abolishment? That means if meiosis is abrupted, if meiosis is stopped in yeast. Why it happens? Because the meiosis specific RNA is degraded. If MI dots are absent, it is a good thing because it will not degrade the DRS. If DRS are stabilized, then meiosis should occur because meiosis is dependent on DRS containing RNA. Therefore, when DRS containing RNA are degraded, then meiosis will not take place. Is it clear to everybody? DRS containing RNA is responsible for meiosis, and DRS containing RNAs are degraded with the help of MEI dots. Is it clear now? How B
Yes,
is the answer? Okay, fine. Next question. Which of these are always expressed? Regulatory non-coding RNA, housekeeping non-coding RNA, meiotic RNA, or mitotic RNAs? We have just discussed this. For 1 minute and then I will switch the screen. Okay. Okay, so okay. So Janara has answered B, Kusuma has answered B, Kavita has answered B, and Renuka has answered B. And what is B? Housekeeping non-coding RNA. Yes, it is the correct answer. Just a minute. Yeah. So housekeeping non-coding RNA is the correct answer. Next question is, a non-coding RNA that is believed to be important in brain development in human is snRNA, tRNA, HAR-RNA, and 28S RNA. So this part, I think this example I have excluded, I think, in today's slide. So the correct answer is, okay, I'll just wait for 1 minute and then I'll tell you. I don't know whether you will know or not. So I think you might be studying about this when your other TA will might explain you this question. The correct answer is HAR-RNA. There is one example which is given in your this thing in your class. I think you can just go through it. I have not included that. There is a paper. So sir has given you a paper to read. So in that paper, there's a description about this HAR-RNA, and there is some little bit description in the slides also. So it is in the lecture sixth lecture. It is there. So you can just go through the sixth lecture. Anyways, your other TA will explain you this question, hopefully, in her class. Okay. So Sky complex facilitate pre-mRNA splicing. True or false? Sky complex facilitate pre-mRNA splicing. True or false? 1 minute to think and then I will switch the screen. Nobody has attempted this question? True. Okay. Others? True or false? It should be true or false. Okay, true. Satyashri is also saying true. Okay. So the correct answer is false. Why? Because key complex is responsible in, it functions in cytosol. That means at the level of translation. That means it is obviously post mRNA splicing. So how it can be pre mRNA splicing then? It will obviously be post mRNA splicing, no? Because key is functioning in the cytosol. It is a, we have studied that key, there are two kinds of endosomal complexes, exosomal complexes. First is TRAMP complex, which functions in the nucleus, and key complex, which functions in the cytoplasm. So in the cytoplasm, if it is functioning, that means the mRNA, it has, when it is coming in the cytoplasm, that means it is about to translate. So if it is about to translate, obviously it has gone through the mRNA splicing and everything. So it is false. Is it clear? Is it clear to everybody? This the concept of ski.
Ma'am, it is facilitated pre-tRNA splicing.
Sorry, it facilitates
Pre-tRNA splicing.
pre-tRNA splicing. So, pre-tRNA, I think you have to go through and see, because up till now, what we have studied about pre-mRNA, we are mostly focusing about pre-mRNA. But always remember that whenever it is occurring in the cytoplasm, that means whatever kind of RNA it is there, whether it is an mRNA, whether it is a tRNA, so the processing of RNAs, the formation of a complete mature RNA, it always happens in the nucleus. So whether it is a pre-mRNA or pre-tRNA, if it is not completely made, it will not come out of the nucleus. So it will never be pre, it will always be post only. Only those processes that are occurring in the cytoplasm, that will be affected by Sky complexes, not the processes that are occurring in the nucleus. Because Sky complex does not occur, does not works in the nucleus, it works in the cytoplasm. So if it is a pre-tRNA, that means the tRNA is not completely mature. If it is not completely matured, it will not come out of the nucleus. And if it will not come out of the nucleus, that means ski will not work over it. Okay. Okay. Next question is again of ski. So you can understand that this ski proteins are very important. So many questions are coming. Ski is known to recruit dash enzyme for mRNA degradation. Endonuclease, 3' to 5' exonuclease, 5' to 3' exonuclease, or nuclease. 1 minute to think, and then I will switch the screen. Okay, let's see. False. B. Oh, sorry. B. Janara is saying B. Renuka Devi is saying C. Okay. So the correct answer is B. Why? Because ski 7 is a kind of, is a complex of exosome, and we know that exosomes, it works at the 3' end. So it would be 3' to 5' exonuclease. At the 5' end, XRN1 comes. So it works in 3' to 5' direction, because ski 7 will bind to the 3' end. Okay. Clear is it? Is it clear? Renuka, is it clear? Okay, fine. Now see the next question. Exon junction complex causes nonsense mediated decay. True or false? Exon junction causes, exon junction complex causes nonsense mediated decay. True or false? True. Janera is saying true. Okay. Other? Okay. So the answer is false. Why? Because nonsense mediated decay is not caused by exon junction, it is caused by the distance of stop codon from the exon junction. Okay. If you remember, we have studied in this slide. See, if the distance is more than 55 nucleotide, if the distance of the stop codon from the exon junction is more than 55 nucleotides, then it will lead to nonsense mediated decay. If the distance is less than 55 nucleotide, then it will not lead to nonsense mediated decay. That means it is not just the exon junction complex that causes the nonsense mediated decay, it is the distance of the stop codon from the exon junction complex. Okay, it is the distance of the stop codon from the exon junction complex. Now, next and the last question for the week is, non-coding RNA are always stable. True or false? Non-coding RNAs are always stable. True or false? Let's see. Okay. So the correct answer is, again, okay, anybody wants to say anything?
False.
False. Yes. So non-coding RNAs are not always stable. False. Okay. So with this, we have completed our today's lecture. So first, we have studied the different mechanisms of RNA decay. It can be de-adenylation dependent, de-adenylation independent, endonuclease mediated. If it is important point to notice that at 3' end exosome comes and degrade, at 5' end xRNA comes and degrade, and the decapping enzymes are DCP1 and DCP2. Then we have studied about independent and endonuclease mediated decay. Okay, then we studied about the p bodies. P bodies are nothing but it is made up of the translationally repressed RNAs and the proteins which are responsible for the mRNA protein. So these are granules that contains the repressed RNA and decay, and the proteins that decay the RNA. Then we studied about the exosomes. That exosomes, they can function both in nucleus as well as in the cytoplasm. The naming of the exosomes follows like RRP41, RRP. If RRP is present, is written anywhere, always remember it is exosome only. Then we see that tram complex works in the nucleus, whereas sky complex works in the cytoplasm. In the cytoplasm, there are three pathways. First is nonsense mediated decay when a premature stop codon comes. Okay. Then there is non-stop decay when there is no stop codon or the ribosome is not stopping. And then there is no-go decay when there is a secondary structure due to which ribosome is stopped or halted. Okay. Then we studied about some kind of autoregulations. Autoregulation, here we studied that of NAB protein, that is poly-A binding protein. We studied the degradation of the meiotic transcript in the mitotic cells, and we studied that how the activity of exosomes regulate the expression of gene. And then we started with some non-coding RNAs, microRNAs. Okay, so with this we finished our today's class. So hope you could understand anything from it. Even if you are able to understand at least 0.01%, it's more than enough for me. But because it is a little bit more complex, so I would suggest you to again go through the lectures, read it, reread it, read my PPT also. You can go through my lecture also. It will be available by tomorrow morning by sure, because I have to do
Ma'am,
some work.
non-coding RNAs are not regulated, true or false, ma'am?
Non-coding RNAs, see, the question was, see, the question is, non-coding RNAs are always stable. No kind of
ఎక్కడ
RNAs are always stable. Each and every kind of RNA, whether it is a coding RNA or non-coding RNA, all the RNAs are prone or marked for degradation at some or the other point, because the function of RNAs is context dependent. If the context is not there, the RNA would be degraded. So non-coding RNAs are always stable. False. Okay.
Okay, ma'am. Thank you.
Okay. So any other question? So just go through the lecture, and if you have any problem, you can ask me in the next class. Okay. Kanchana, you want to say anything? Okay, fine. Okay, then we'll end today's session. Chalo then. Okay, we'll meet again next week. Bye everybody.